So, what is cloning?
Well, cloning is a whole bunch of different processes that can be used to make copies of a biological entity. The copy material, which has the same genetic makeup as the original, is referred to as a clone. Scientists have cloned a whole bunch of different biological materials, including genes, cells, tissues, and even entire organisms, such as like sheep.
How many types of cloning are there?
There are three different types of cloning. There's gene cloning, reproductive cloning, and therapeutic cloning. Gene cloning makes copies of genes or segments of DNA. Reproductive cloning copies animals. Therapeutic cloning involves cloning embryonic stem cells for experiments aimed at creating tissues to replace injured or diseased tissues. Gene cloning, also known as DNA cloning, is a very different process from the other two. Reproductive and therapeutic cloning share many of the same techniques, but are done for different purposes. Researchers routinely use cloning techniques to make copies of genes they wish to study. The procedure consists of inserting a gene from one organism, often referred to as a foreign DNA, into the genetic material of a carrier called a vector. Examples of vectors include bacteria, yeast cells, viruses, or plasmids, which are small DNA circles carried by bacteria. After the gene is inserted, the vector is placed in laboratory conditions that prompt it to multiply, resulting in the gene being copied many times over and over.
Well, what is therapeutic cloning?
Therapeutic cloning involves creating a cloned embryo for the sole purpose of producing embryonic stem cells with the same DNA as the donor cell. These stem cells can be used in experiments aimed at understanding disease and developing new treatments for disease. To date, there is no evidence that human embryos have been produced for therapeutic cloning. The richest source of embryonic stem cells is tissue formed during the first 5 days after the egg has started to divide. At this stage of development, called the blastocyst, the embryo consists of a cluster of about 100 cells that can become any cell type. Stem cells are harvested from cloned embryos at this stage of development, resulting in destruction of the embryo while it is still in its test tube. Have you heard anything wrong because of cloning? I heard that reproductive and therapeutic cloning raise important ethical issues, especially when the use of these techniques are used with humans. Reproductive cloning would present the potential of creating a human that is genetically identical to another person. This may conflict with long-standing religious and societal values about human dignity, possibly infringing upon principles of individual freedom, identity, and autonomy. Some see human cloning as a way to avoid passing on a deleterious gene that runs in the family without having to undergo embryo screening or embryo selection. Therapeutic cloning would require the destruction of human embryos in the test tube. Sadly, some people argue that using this technique to collect embryonic stem cells is wrong, regardless of whether such cells are used to benefit sick or injured people.